This is Dr.K.Manimegalai, Assistant Professor, Department of Zoology, Sri GVG Visalakshi College for Women, Udumalpet. So in microbiology, let's see a small topic about dairy microbiology. So what is a dairy microbiology? So this is the study of milk and microbes present in the milk products is called dairy microbiology. So what are the microbial examination of milk and milk products? So number and the type of bacteria present in the milk that can be estimated by the bacterial examination. So this bacterial examination, it helps to determine the effect of sterilization. The raw milk always contains 500 to several millions of bacteria per milliliter. So there are various methods used for bacterial examination of milk or judging the quality of the milk. Let's see a small introduction about milk. So what is a milk? It is a compulsory part of daily diet as for mother as well as growing children. The microorganisms present in the milk is Lactobacillus, Enterobacter, Pseudomonas, Bacillus and Klebsiella. And the raw milk can be originate from or form of different sources such as air, milking equipment, feeding soil, fecuses and grass. So these are the contaminations of reasons. So milk is a complex biology fluid and it is a phytochemical properties of milk is a good growth media for microorganisms. So this is the bacterial examination of the milk. And what are the tests? The first test is detection of developed acidity. The two tests coming under this clot and boiling test, alcohol test, standard plate count, direct microscopic count, reductase test, phosphatase test, turbidity test, an examination of the specific pathogens. It is for test for coliform bacilli, test for tuberculin, and test for tuberculin and the test for the brucella. So these are the microorganisms responsible for the test for coliform bacilli. So in this test, the McCann-Key fluid medium is used for the protection of the acid and gas after incubation. So this test gives an idea of possible fecal contamination. This test having the responsible for the contamination of coliform bacilli bacteria either come from the dust, dirty utensils and dairy workers. So what are the types of bacteria in milk? That is the acid forming bacteria, alkali forming bacteria, gas forming bacteria, proteolytic bacteria, inert bacteria and human breast milk. And what are the pathogens present in the milk? That is the E. coli, Streptococcus pyogenes, Mycobacterium bovis, Bacillus anthracis, Salmonella species and Brucella species. So this is the importance of milk examination of pathogens. It is consumed by man. Milk is further used for obtaining many milk products like cheese, creamy butter, ice cream, ghee, etc. And this is the direct microscopic count method. This is the very important method in bacterial examination of milk. So in this method, 1.0 centimeter area is marked on the glass slide and 0.1 ml of milk is spread uniformly over this area and the milk is dried and stained. The stained preparation is examined under the microscope. So we have to examine the direct under the microscope, the number of bacteria will be counted. So this is the direct microscopic count procedure. And next to the direct microscopic count, it is the plate count. Here the single isolated bacteria can be visible and isolated colonies. It can be determining the number of colony forming unit, that is CFU. And next to this is the calculation coming under the CFU. Number of colonies in the plate divided by dilution of sample into the volume plated in the sample. So equal to number of bacteria per ml. This is the calculation coming under the count of the cell. So these are the biological tests used most often for the standard plate count, that is the serial dilution techniques. So dilution 1 to dilution 10, it will be continued. So dilution increases, the microorganisms will be decreases. This is the colony counting. So we can count the colonies on each plate. Select all the Petri plates containing between 30 and 300 colonies. And what are the pasteurization of the milk? That is, LTH is low temperature holding method, HTST is high temperature short time method, UHT is ultra high temperature. The benefits of pasteurization of milk is it kills and removes all the pathogens and kill most non-pathogens and thermophilic organisms. And this is the dye reduction test using dye is methylene blue or reazarin dyes. 0.5 ml of dye is added to the 5 ml of milk and incubate. After that, if there is any change in color, it means high bacterial load. It is not a good quality. If it is no change in color, it having low bacterial load, having good quality. And standard plate count. So this is the determines the total number of bacteria to form a countable colony. An agar plate of 1 ml of milk is incubated aerobically at 90 degree Fahrenheit for 48 hours. So determination of viable bacterial count, it also by 4 plate method and methylene blue reduction test, methylene dye is used. Thank you. So that's all about the bacterial examination of milk.